All right, good afternoon. It's 12 o'clock on Wednesday. That means it's time for the Iowa Learning Farms Conservation Webinar Series. Matt Helmers with Iowa State University, fortunate to be able to moderate the webinar today. As a reminder, Iowa Learning Farms is celebrating 20 years this year, so be on the lookout for special events that might be coming up. And we're really fortunate to have today Dr. Bryan Emmett, that's a research biologist with the USDA Agricultural Research Service. He's with the National Laboratory for Agriculture and the Environment. For those like myself that have been around a few years, that's the lab formerly known as the Tilth Lab, Soil Tilth Lab here in Ames, Iowa. And so they really have an all-encompassing research lab there doing work on many different areas, including the microbiology. So Dr. Emmett is going to talk to us today about partners below ground, opportunities for management of plant fungal symbiosis. As always, if you have questions, please type those in the chat box and we will get those to Dr. Emmett at the end of his talk. With that, Bryan, the virtual floor is all yours.
Great. Well, thank you, Matt, and thank you for the invite to talk today. I'm excited to be here. My research within ARS broadly is focused on nitrogen and nutrient cycling and how soil and plant microbe interactions shape that. And within that context, one group of organisms that I particularly enjoy thinking about and working with are mycorrhizal fungi. And so today I thought I'd share a bit about their ecology and the management effects that we have on them in the field. This isn't a prescriptive talk about a particular management practice that you can maybe take away, but more some ways of thinking about soil biodiversity in the context of arbuscular mycorrhizal fungi and how that might shape our management choices. So AMF are obligate symbionts. They're one of several types of mycorrhizal fungi. They form associations with about 80% of plant species. And they're known as endomycorrhizal fungi. And because as they infect the plant root, they grow into the cortex of the plant and actually invade the cell wall of the cortical cell. So in the upper right-hand corner, you see a fungal membrane that is enveloped within the plant cortical membrane. And so the membranes are intact, but the fungus is inside the plant root. And this structure is an arbuscule. That's what we get the name from. And it's this tree-like structure that is made to maximize surface area, an exchange area between the plant and the fungus, really. So the fungus is providing nutrients for the plant across this membrane, and the plant's providing energy, primarily in the form of lipids to the fungus. So we're trading carbon for nutrients, essentially, in soil. But then the fungus grows back out into the soil. The extra radical hyphae, that's the hyphae that's outside of the plant root, is there foraging for nutrients, and it's extending the plant root effect essentially away from the root and into the soil, affecting the soil ecosystem. And this can be quite severe. Up to 20% of fixed carbon can actually enter the soil through this mycorrhizal pathway. And it's here that mycorrhizal fungi then interact with soil microbial communities, forage for nutrients, etc. So this is really, it's an ancient symbiosis. These organisms evolved around 500 million years ago. They're thought to have associated with the earliest of land plants and maybe even forming the first proto-roots. And the symbiosis between between land plants and these fungi is really the ancestral state of things. And so this symbiosis has been lost in several lineages since then. But like I said, most plants are able to form this symbiosis. Brassicas are one notable agricultural plant that can't. Now, within this evolutionary history, AMF also lost a lot of the enzymes that they need to degrade organic matter and live on their own. And so they're dependent on the plant host. And the plants have evolved in symbiosis with them. Now, a couple of things to note. because they're dependent on the plant host, we can't culture these organisms independently of the plant. So we have to set up co-culture systems. And what this means is even though we have this rich evolutionary history, is we know quite little about the functional diversity of AMF, or we can appreciate quite little about the functional diversity of AMF that's derived over the last 500 million years. We have some hints, but there's a lot that we don't know because we can't manipulate the organisms independently of the plants. The second thing I just want to note is if you're a grower and you're thinking about managing soil biodiversity and you're thinking about AMF, that this evolutionary history is sort of an inheritance that we have and kind of an awesome system in my mind to be working with. So I always like thinking about these organisms in that context, that sort of ancient context. So these organisms do provide pretty diverse plant and ecosystem benefits. Nutrient exchange is sort of the salient characteristics, and particularly for nutrients that are less mobile in soil. So phosphorus is the big one. It's much more efficient for the plant to give carbon to the fungus and for the fungus to grow its hyphae out in the soil looking for phosphorus and transporting it back to the plant root than it is for the plant to build a root system and fully explore that space. There's also micronutrient exchanged involved, as well as nitrogen. And even though nitrogen is a relatively mobile nutrient in soil, there are still situations where it becomes important. And the nutrient exchange is important up to 70 to 100% of phosphorus uptake by a plant can come through this mycorrhizal pathway, even in really fertile soils where phosphorus is readily available. But it's not limited to nutrient exchange. AMF are known to increase plant tolerance to biotic stresses, pathogens, drought are big ones, heavy metal toxicity. And then there's a number of ecosystem services that they perform as well, kind of in the context of soil health that are nice to think about. They're agents of soil aggregation and stabilization, certainly redistributing organic carbon into the soil in very close association with the soil mineral phases. And through that, changing soil water holding capacity. So a pretty wide range of potential benefits. It's worth noting just a little bit about the soil structure. I'll come back to this a little later in the talk. So the mycorrhizal fungi are both helping build soil aggregates and protecting those soil aggregates from disruption. So the hyphae themselves, shown in the micrograph on the left, are in really close association with these aggregates, enmeshing them, binding them together. Extracellular polymeric substances on the surface of the hyphae are helping to bind particles together and lead to organic mineral associations. And the effect that we see in the field and in soil is that there's both an increase in total aggregate stability when we measure that in the presence of AMF, but particularly we see aggregation into larger aggregate sizes. And so there's a shift from smaller sized aggregates, and in the presence of AMF, we get more of the larger diameter aggregates that are over 250 micrometers in diameter. So a symbiosis is two organisms that are living together. But for that symbiosis to be a mutualism, both partners have to benefit. And evolutionary and ecologically, this is a balance, as really both of these organisms are trying to maximize their reproductive success. So in a simple sense, the degree to which plants are benefiting from AMF depends on ecosystems factors such as the nutrient status of the soil, abiotic stressors, disturbance regimes, all these factors that change the balance between the resources that the fungus is providing, or the stress tolerance that the fungus is providing, and the carbon that is allocated to the fungus, which is a cost for the plant. Now, Nancy Johnson did some pioneering work back in the early 1990s, where she looked at prairies that had been fertilized and unfertilized, and she collected soil from those and AMF communities from those, and then re-inoculated to test plant response. And what she found was that under a history of fertilization, we had actually selected for sort of a parasitic phenotype over time. So AMF from the fertilized background contributed less to plant growth under a range of nutrient statuses, and also even could negatively impact plant growth. Whereas AMF from the unfertilized, at least it didn't negatively impact plant growth, and in certain situations, positively impacted it compared to the non-inoculated controls. And so this is sort of at a selection pressure, sort of changing the balance and phenotypes that we see in the field. I don't want to give the impression that fertilization itself is an issue. And this is a prairie system. It's not immediately translated to agricultural systems. We can think of this better, not as a fertilized versus unfertilized system, but maybe where the balance of nutrients is off, right? In a system where they're not being removed from the field, the fertilization is leading to increased nutrient saturation. If an agricultural field's in balance, these same selective pressures may not be applying. Now, what she also noticed, sort of going along with this decrease in plant performance, was that the phenotype of the fungi themselves was shifting to where there was greater allocation on the fungus's part to reproductive structures, spores, storage features, they're known as vesicles. And then because of that, sort of less transfer of nutrients, phosphorus in this sense. And so we sort of selected for a more aggressive phenotype over time. And Miranda Hart's group, thought about this and asked the question of, if we're culturing these organisms, either in the lab or for production of inoculum, and here I do mean in a co-culture system with the plant, how is that selective pressure changing the fungal phenotype? And what they found was that it can have some of the same effects. And so repeated in vitro culture with the plant could lead to a fungal phenotype where that allocated more resources towards reproduction and less towards providing plant benefit, which is just something that we need to keep in mind as we're thinking about inoculants, and certainly the companies themselves need to as well, as how to maintain the plant benefit under those situations. So that kind of leads me to the question is, can we manage for this symbiosis? We suggest it's important, but what can we do in the field? And the first stab at that is going to be through, inoculation. Can we bring the fungus in and sort of increase its activity? And while it's fairly difficult to observe in any given study, when we look at a meta-analysis, sort of a preponderance of studies over time, the top graph is the effect size in any given study. Sometimes we do see a negative response. Somewhat frequently, we see a neutral to no response, but there's also quite a few times where we see this positive response, and that's the preponderance of studies themselves. And these are in grain crops, so these are systems that we're used to thinking about here in Iowa: corn, soybean, wheat, barley. Actually, I should say barley is one of the few crops that didn't show a positive response. We're not quite sure why that is, but that's suggesting that there's still benefit to be had in our agricultural systems for an inoculation treatment. There is some context dependency to this, and so the greatest benefits were shown where there was no fertilization, and particularly where there was no fertilization of phosphorus, though I will say that there were still positive benefits observed even when there was fertilization with phosphorus. And we can think, too, about sort of what's limiting for the plant, not just whether or not it was fertilized or not that year. But in a collection of studies, if we look at the tissue nitrogen to phosphorus ratio, in those systems that were most phosphorus limited, the previous authors have seen the most benefit from inoculating with the fungus. And so certainly how we manage nutrients in these systems, I'm not suggesting that we don't fertilize, but if we're looking for a benefit from these symbionts, thinking, do we need as much? Do we need to maintain as high of levels of that nutrient in the soil to get the same plant growth benefits? Maybe one opportunity for taking advantage of this symbiosis. So Along this vein, a group of researchers in Switzerland conducted on-farm inoculations at 55 different sites. And again, they observed a sort of expected distribution where there were some fields that really responded to the inoculation treatment, others that didn't, or others that had even suppressed growth following inoculation. And so if you're a grower, you really want to know if you're one of these fields that can benefit from AMF, or if you do not need to spend the money, or it might even be detrimental. So they collected a lot of covariates, sort of looking for what indicators there were to predict inoculation success. Interestingly, what they found was that microbiological indicators were the best predictor of the growth response. And they found a handful of fungal lineages is that when they were present or abundant in the field, predicted inoculation success as measured in a greater growth following inoculation. And so this suggests that the resident community is somewhat important for how this inoculation goes. Now, notably, a lot of these lineages are associated with pathogenesis. It's hard to say that they are pathogens because there can be a lot of functional diversity within these groups. But it might be a hypothesis that the benefit that they observed here really had to do with biotic stress resistance in this context. So maybe just to wrap up that thought, I think we're a ways from, a test that we can give to growers and say, hey, you have these microbes, therefore you should inoculate. But I think it does point a route forward for, as a research community, can we identify, a usable test that can provide growers with the information that they need to inform a decision of whether or not to inoculate or not as they go forward. Now, on a more sobering note, a lot of these tests are done with lab strains where researchers are used to working with them and can confirm that they're viable. This same group, which is a very well-respected group, tested 25 commercial inoculants independently at 2 labs, one in Australia, one in Europe, and there were some field inoculations that I'm not showing here in the United States. And somewhat troublingly, what they found is that out of 25 commercial inoculants, 84% of them did not contain viable propagules, or at least propagules at the abundance that were needed according to the labels. And so the very tall bar on the left is a control fungus from the lab that was doing this research. So the inoculations work in this context. This is a greenhouse assay. But a lot of the commercial inoculants didn't achieve that success. The picture wasn't nearly as bad in Europe, and so there may be a transport or handling issue here. I'm not quite sure what's happening, but still there was several products that were no longer viable. And so for an industry interested in mycorrhizal inoculants, and this industry is growing, it's present, it's growing, the authors put forward sort of a framework for thinking of how can we manage the quality of these inoculants in a way that provides a surety that we're getting a viable product and that it's a product that's well matched for the environment. And I think this is going to be really essential if the industry is going to take off. So inoculants are one way of managing for these fungi, but what about field management itself? I'd argue that everything we're doing in the field has the potential to impact soil biodiversity. And so can we understand how to harness these communities in a way that may not require introducing another inoculum? And so for this research, I'll share a little bit of what we've done at one of our long-term agroecosystem research sites here in Ames, Iowa, where we're comparing alternative management practices, mainly focused at sustainably intensifying the system. So this occurs right outside of town here. And there's 4 systems that I'll be comparing, and I think they're rather nice to evaluate in their impact on mycorrhizal fungi because they have rather contrasting potential impacts. Sort of our standard basic practice is a corn-soybean rotation that has chisel plowed after corn and no cover during the winter or the non-growing season. And then We have a no-tillage treatment, so now we've removed that mechanical disturbance that can disrupt the hyphae. And we have a no-till treatment with a rye cover crop, so now there's no physical disturbance, and there's a winter host following both phases of the rotation that's present year-round. Between the cash crop and the cover crop, there's a host present year-round. We also have a winter camelina relay crop. And so winter camelina is a brassica. And I mentioned before that brassicas do not form association with AMF. And so now we have those carbon inputs during the non-growing season, but we don't have the host for AMF. And so how does that affect both the abundance and activity of these fungi and maybe some of the ecosystem properties that we are interested in? And so we can sort of arrange these treatments on an axis of, the quality of AMF habitat, versus the carbon inputs, and think about how that might impact soil structure or soil health. And we could hypothesize that these non-growing season management systems are going to alter the composition and maybe the seasonal dynamics of these plant-associated organisms, and particularly that arbuscular mycorrhizal fungi abundance and a measure like aggregate stability would be higher in systems with less disturbance in a winter plant host. So we went out to this field 5 times over the growing season in 2021, collecting soil from plant roots, collecting the plant roots themselves, collecting soil from the rhizosphere, and soil from the inner row as well, so that we can see how these dynamics are unfolding in sort of different compartments in relationship to the plants. And that first hypothesis, well, maybe the second hypothesis, that AMF abundance will be higher in less disturbed systems with a winter host. If we look at the activity on the root system, this is partly bared out. So the systems with less disturbance and a winter host, the no-till and the rye cover crop are the rye cover crop's the purple box. At the beginning of the season, we do see higher activity on the root system, but it's somewhat marginal. And we're ranging between 40% root colonization and an average of 60% root colonization in our sort of best and worst treatments. And that's not a huge difference in terms of activity for AMF. So we have a fairly robust population of AMF, even in the systems that we think we're going to be less favorable to them. And as we move away from the early harvests, so the first harvest took place 1 month after planting, and then each harvest is a month subsequent, as that effect really disappears. And so the activity on the root system is kind of the same through most of the season, regardless of the system that we're evaluating. When we move to soil, though, and now we're measuring a lipid biomarker of AMF, we see a kind of dramatic difference. And here, that rye cover crop, the system that has the least disturbance and the winter host, starts off with a high proportion of AMF biomass at the beginning of the season, and that difference is sort of maintained throughout the entire growing season. And the no-till system, where there's no physical disturbance, initially starts with lower AMF biomass in the bulk soil, but over the course of the season recovers to where that fungal hyphal network, you can envision it, is sort of recovered and is the same by the end of the season. And so when we're managing these systems, we might not be managing so much for colonization and activity on the roots themselves, but rather for the persistence and function of that hyphal network within the soil. And that can be important when we think about that nexus of soil health and the activity of these organisms. And so here we're looking at aggregate stability. And it's these less disturbed systems, the no-till and the system with the rye cover crop, which have both the highest proportion of stable aggregates and, as I hinted to earlier, a larger mean weight diameter. So we have more aggregation, but also more of the larger size class. And, these are association of treatments. If we're looking for whether or not the abundance of AMF themselves are associated with these factors, if we try to correlate this biomarker with aggregate stability, we do see a relationship between the two, But we primarily see that at the beginning of the season. So the graphs on the left are correlations of aggregate stability and AMF abundance at that first harvest, and the ones on the right at the last harvest. And it's at that first harvest where we see the correlation. And so this suggests that either A, this is most sensitive indicator, if we're looking for biological indicators of soil health, and then, or B, if we're trying to manage for aggregate stabilities, for soil structure, that maybe we should focus on, the relationship of AMF early in the season and select for systems that are more stable in the abundance and activity of these organisms over time. I will note we sequence the DNA in the roots in the soil in the rhizosphere, and so we can look at how the composition of AMF changes over time. I don't have time to really dig into this yet, but we don't see a dramatic shift in the composition of the AMF community. It's more of a question of abundance. And the other thing I want to note is that there's incredible diversity of AMF. Even within these agricultural soils, there's still quite a diversity of organisms that are there and that we have to work with. So this is just one site here in Iowa. And I'm also part of a cross-site research network led by Veronica Costa-Martinez and her postdoc, Wayne Roper, within the Agricultural Research Service. And so they've been collecting soils from across the country, 14 sites, to assess sort of soil health metrics and how they relate to each other. And if we look at this AMF biomarker again and our outcome of soil aggregate stability, we see this positive correlation. And there's a lot of variation around that line, but AMF abundance itself explains about 20% of the variation in aggregate stability across all of the samples from this network. And what I think is encouraging, kind of in line with our previous results, is that we can manage for them. So these are experimental sites with different treatments. And in the same way that the data that I showed you from Iowa, where we add tillage, we see a decrease in AMF abundance in soil. Where we add a cover crop or we add crop biodiversity, we see an increase, and even a slight increase with manure. So AMF abundance is related to soil structure across these sites, and it is responsive to management. So we do have some tools with which we can shape these communities. And so I'm going to end it there. Here are some of the references that I covered if people are interested. And I hope I've been able to give you, some idea of the role that these organisms can play and some indication of how we might think about managing them as we move forward. So I am looking forward to your questions. First, I'm going to pass it back to Matt to provide some logistical
items. You could take a drink of water and I'll do the logistical items. So if you watch this webinar live and you want to earn a CCA CEU, please email your name, the name you entered to watch the webinar, and your CCA number to Elena Whitaker, ALENAW @iastate.edu. Again, ALENAW @iastate.edu by 5 o'clock today. So 5 o'clock Wednesday the 7th, ALENAW @iastate.edu. Next slide, Ryan. If you watch this webinar live and are willing to fill out a voluntary demographic survey, we'd be most appreciative. Only one survey response is needed for all 2024 webinars. So if you filled one out already for the calendar year 2024 webinars, thank you very much. If not, and you're willing to do so, we'd greatly appreciate it. You can scan that QR code. I'll keep talking delaying so anybody can scan that. Otherwise, you can go to iowalearningfarms.org/survey, iowalearningfarms.org/survey. Okay, and then to just put in a plug for the next webinar, we're going to have wood chip bioreactors, expanding carbon sources and implementation strategies. So a colleague of mine, Dr. Michelle Sapir here in the ABE department will be talking. Also, I'll put a little plug in tomorrow. There's going to be a virtual field day on some bioreactor information with, I think, Dr. Billy Beck and Dr. Michelle Sapir, 1 o'clock, if I'm not mistaken. Hopefully Mitch or Ann can let me know if I'm wrong about that. Okay. We do have some questions coming in. So quite a few questions coming in, Bryan. That's great. We'll start with across the LTARs at low AMF abundance, aggregate stability can be high or low. So maybe the positive relationship is a coincidence. And that was question mark on that last part.
Yeah, so that's a great question. I think there's a few things that are happening in these global data sets. One is our site's sort of natural soil features. There's no edaphic factors that may disfavor AMF, maybe the same ones that are also leading to less soil aggregate stability, right? So we might have sites with just inherent soil properties that are not going to lead to as great of aggregation. And so there is a coincident issue there as well. But I think it's powerful that the same management tools that we think of as improving soil health are similar management tools to those that are supporting these organisms. And we do have a mechanistic basis for thinking that these organisms are positively impacting soil structure, right? The same way that a living root improves soil structure, AMF do as well. And so, one thing that you picked up on is in our cover crop treatment, we can't really distinguish what the effect of the AMF in that treatment is versus just the fact that there's a plant root there all year round as well. But they're not mutually exclusive, right? So we're managing for sort of an ecosystem state here.
Here's the next one. What level of specificity is useful to consider for practical applications? The actual impact of AMF can vary based on the species and variety of both symbionts, but should farmers and crop advisors be thinking at that level, or stick to more is better?
Yeah, that's a really great question. So at some level, I'm not sure more is better. And I was trying to hint at this, and maybe it wasn't super clear. We actually get a fair bit of root length colonization in all of our treatments, right? And so the question is like, what is that providing in terms of function? And we could certainly provide inoculant and boost that early in the growing season. But would we be shifting what that fungal hyphal network is in soil? So I think more of what is important.
Okay.
I believe. So the second part of your question was about functional diversity in AMF and how much is important. There's a few things that I shared to suggest that it is quite important. Some things that I didn't share is how much intraspecific variation there is. Strains of the same species, even being cultured in the same lab, can have functional variation within them. And so our ability to discern what this broad phylogenetic diversity within AMF means for management or means for plant benefit is poorly resolved because these organisms are difficult to work with. And within a particular species, I'd say we're even more poorly resolved. But the functional work that has been done suggests that we can shift them with our, the Nancy Johnson's work. We can alter the benefit that these organisms are giving based on how we set up the system. So to summarize, I wouldn't say more is better. Sort of the quality and how they fit into the performance of the system becomes really important.
Okay. Next one, do you think it's worth including a test for AMF in analytical soil health assessments that might already be including aggregate stability?
Yeah, that's a good question. So some of Veronica Acosta-Martino's work is looking at just at this, right? It's like, can these fatty acid methyl esters, which is the biomarker we're using as a surrogate for AMF biomass, are they not a pretty good indicator of soil health? So, what indicator? This is an interesting philosophical question. Is like, what makes a good indicator? Something that's easy to measure and reproducible and is hopefully sensitive to management, right? So I don't think of them as mutually exclusive. Um, and so I wouldn't say that, AMF make a better indicator than soil aggregate stability. Um, both can be difficult, although FAME, I'm starting to believe that FAME could be cheaper to run than
Okay.
soil aggregate stability, because soil aggregate stability can be rather labor intensive depending
Mm-hmm.
on how you run it. So, I think they're complementary. And if we're trying to boil it down to one function, that's a tough question. I guess I'm hedging. I'm not giving you a very
Yeah,
clear
no,
answer.
that's that's okay. That's your prerogative, right? There's kind of another testing question is what are your thoughts on the value of soil tests that use genomic sequencing, such as trace genomics?
Okay, so I'll start by saying I'm not fully aware of the what trace genomics offers,
Okay.
or really any commercial soil testing that uses genomics information. So I can't comment on its utility entirely. Um, from my perspective, we have we still have a lot to learn about what some of the sequence data means, how it relates to whether you're thinking about soil health or predicting crop performance. There's a lot of promising developments in this realm. Um, I'll leave it there.
Okay. Here's another good one. So, what do you believe causes the management practices to degrade the AMF? For instance, tillage, does it physically disrupt the AMF? Does it cause a chemical state imbalance? Or, yep.
Yeah, I think that's exactly right. We think of it as the mechanical disturbance, right? So, there's a hyphal network in soil, and when we till it, we're essentially breaking that up. Some of those fragments will regrow and reinfect a plant, and there's certainly spores or there's root fragments from which that hyphae can regrow, but we've disturbed the system, and so now to remake that hyphal network, it has to reinvest to build that biomass. Um, as well.
Okay. Uh, okay. Do I guess yeah, this is do any of the chemicals that we apply or different seed treatments or anything like that, would those impact AMF or have they been shown to impact AMF?
Yeah, so I'm not as familiar with the seed treatments to be able to comment on it. I was trying to find out about sort of the herbicides and if they were having non-target effects. And there's certainly some literature that suggests, for fields that we're spraying herbicides on, that AMF abundance is suppressed. I could not figure out if that is a direct effect, like it's being translocated and having an impact on the fungus, or if we're just, if there's a field treatment and we apply herbicide to it, there's less of a host, and therefore there's going to be less AMF. So we are impacting the fungus. I don't know personally, what is direct versus indirect in terms of that. It's something I'd like to know in the context of the rye cover crop, right? We measure that the biomass is there. We're assuming that it's still functional even after we terminate the biomass with an herbicide. So that's an assumption, and
Okay.
uh, yeah.
Okay. All right. I think that gets through most of our questions. So, Bryan, thank you very much for this really interesting presentation. For me, a little over my head at times, but that's okay. I learned something new, so I'm going to be on the lookout for this a bit more. Again, if you need a CCA CEU, please email Elena Whitaker, ALENAW, at iastate.edu by 5 o'clock today and put in a plug for next week, Wednesday the 14th, Valentine's Day. All right. What better to talk about on Valentine's Day over the lunch hour than wood chip bioreactors, expanding carbon sources and implementation strategies. So, Dr. Emmett, thank you again for your presentation. I'll give you the last word unless if you have any parting comments.
No, just to say thank you everyone for attending and your attention, and thank you, Matt, for the invite.
Yeah, absolutely. And just as one last point, the archive version, as long as it looks like my recording will have worked, that'll be posted on the Iowa Learning Farms website soon. So if there were any problems, hopefully you can go back and watch the archive version. So thanks everybody and have a great day.